Hello everyone, my name is Daniel Dudek. I'm from the Headwaters Science Institute, and today I'm going to briefly talk to you about statistics, an introduction and application. So, just to start off, what is statistics? The basic definition of statistics is the practice or science of collecting and analyzing numerical data in large quantities, especially for the purpose of inferring proportions in a whole from those in a representative sample. But to break this down, put simply, we are studying or collecting data from a small representative sample of the population, highlighted here in the orange individuals within the large population, surrounded by the whole population, which we did not sample. We take this sample, we analyze the data of that sample, and then we make inferences that tie back to the larger population as a whole. So what we're trying to do is make sense of the population and all of its characteristics, everything, testing claims, testing hypotheses. We're trying to address natural phenomena within the whole population by studying a smaller sample of that population, because it's just not feasible to study every single individual of the entire population. So, why do we use statistics? So some things that we do that are addressed with statistics, meaning things that we as humans tend to do that necessitate the reasoning to use statistics, would be that we as humans tend to be overconfident and jump to conclusions. We may not take the most rigorous approach when assessing a claim, and we tend to be overconfident with that assumption and we think it's just correct automatically. And this can be influenced by prior information that we may have, and we're just sort of generalizing without any regard to the actual data or testing that claim. Coincidences are inevitable, and they're actually less statistically remarkable than people think they are. So coincidences actually occur more frequently than you'd actually think, and you're always going to run into these coincidences. So it's important to address this notion that coincidences are inevitable. It's not something that just happens. Like, things happen by chance, but we want to figure out why they happen by chance. And we also draw patterns when no patterns exist. So this might be one of the most important components or tenets, is that we typically as humans will try and draw patterns when none exist. A pretty funny situation is when people look at the moon and they try and see a face in the moon, or they look at some type of object and they're able to see a face or something of that sort. What their brain is actually doing is trying to find a pattern in the random features of that particular object. And this happens in data as well. Whenever someone is looking at a set of data, they may think that a particular trend is occurring or some type of pattern is occurring, but in actuality, the data is randomly distributed. And there are no patterns in the data. And this is one way, and using statistics is one way that we sort of address this issue of drawing patterns when no patterns exist. So what statistics allows us to do is to make sense of natural phenomena and allow us to test claims. So essentially, we want to have this objective approach using collected data. We want to collect as much data as we can in order to draw conclusions from the data rather than using our emotion to drive these conclusions. So we sort of want to be robotic when it comes to assessing these particular claims and hypotheses. We want to use data, and we want to limit the amount of bias that we have in drawing a conclusion. And statistics allows us to do this. So a few concepts that I want to cover. We're going to cover two. The first one is going to be the measurement of central tendency. So essentially, what a measurement of central tendency is, it's a summary measurement attempting to identify the value in the center of your data distribution. So what this means is you collect a ton of data, but we really want to summarize and find the center point of all of your data that you have. So some of the common measurements for the measurement of central tendency are the mean, median, and mode. So the mean is going to be simply the average of your values in the data set. This is probably one of the most common that all of you have used and learned about. Median and mode as well. And median is going to be the middle value of your data set. So this is exactly right in the middle. And then the mode will be the most occurring value in your data set. And all of these can be used for different purposes. When we think about the measurement of central tendency, if we have a larger data set with numerous values on the extremities, then using the median would be preferred than the mean. And this is simply because the median is less influenced by extreme values, where the mean would be. So if we think about a large data set with, if we think about sort of the income levels in the United States, typically someone is going to refer to the median because you can have a large number of individuals that make a lot of money or an extremely low amount of money when it comes to the income in the entirety of the United States. So the median is less sensitive to those extreme values compared to the mean. So that's one example of when you would use, say, the median versus the mean. Now we want to get into this concept of measures of spread. So if we're looking at basically what the measure of spread is, it's going to be different ways that describe how similar or varied the set of observed values are for a particular variable. And the four measurements of spread that I'm going to be covering today are going to be the range, briefly, quartiles, the interquartile range, and the standard deviation. The standard deviation being of which is extreme importance. So first off, the range is going to be the highest value subtracted by the lowest value in your given data set. So it's going to tell you the range of values that are between those two values. Then with quartiles, so I have an image here of the median and the quartiles. So we can see that the first quartile or the lower quartile, or Q1, is going to be the value that divides the lower 25% of your data and the upper 75%. On the opposite end of this, we have the third quartile or the upper quartile. And this is going to be the value that divides the lower 75% of your data with the upper 25% of your data. And then we have the median, the second quartile or the middle quartile, which is going to be the value that represents the middle of your data set. So this value is going to divide the lower 50% and the upper 50% of your data. And then we have the interquartile range. So this is going to be the collection of the middle 50% of your data. Right there. And so the next measure of spread that I'm going to talk about is the standard deviation. So the standard deviation is a measure of spread of the data around the mean. So this is going to summarize how close each observed data value is to the mean value. So if we have two graphs here, and they're going to relate to each of these scenarios. So on the upper graph, we have a higher standard deviation equaling the variation is going to be high. So if we see here, we have, if these are all of our observed values, and this is going to be the mean of those values, we have a normally distributed data set. The standard deviation is larger compared to this graph, which has a lower standard deviation. And you can see that the observed value, there are more observed values that are variable than the mean. So we have a higher range. We have more variability in the observed values. Now, in this bottom graph, we have a scenario where there's a lower standard deviation or a low standard deviation, and this is going to mean that the variation is low. So the range of the data values observed is going to be lower because it has a lower difference between the higher and lower extremities. And the data is going to be centered more around the mean observed value compared to the upper graph. Now covering hypothesis testing, we rarely use hypothesis testing to test the plausibility of a claim. And we usually break the hypothesis down into two hypotheses. So we have the null hypothesis and the alternative hypothesis. So the null is typically going to be an observation of no difference. This is pretty much the uninteresting part of your experiment, compared to the alternative hypothesis, which is typically going to be the observation of significant difference, or what you are interested in. So in statistics, we're really testing the null hypothesis. We're never really testing what you are actually interested in. It's kind of an interesting concept if you think about it. You know, what we're really doing is generating p-values in order to reject the null or fail to reject the null. So we're really testing the observation of no difference, or the uninteresting part in our analysis. So whenever we're using p-values, we typically, it depends on the field, but a p-value that's less than 0.05, this is going to indicate a significant result, and we are able to reject the null hypothesis. And the p-value and this alpha level is going to depend on the confidence level that your specific field requires for significance. So some fields it might be 0.01 based off of a 99% confidence level, and it really all depends. And then we fail to reject, so that means that the null hypothesis in this situation is pretty much accepted. So we fail to reject it, and this is going to be an insignificant result, and the p-value is going to be greater than 0.05, or whatever alpha level that we have selected. But this isn't to say that, you know, an insignificant result is not good. You know, you can have insignificant results and that, you know, indicate a pretty, you know, good outcome. So just because you fail to reject, that doesn't mean that your experiment is a failure. So going into some important terminology, we have the independent variable, dependent variable, categorical variable, and numerical variable. So first, I want to discuss the differences between independent and dependent variables. So the independent variable is going to be the variable that is typically controlled in an experiment, or your x variable. This value is not going to depend on another variable. It's independent by itself. That's why it's something that you typically would control. Whereas the dependent variable is the variable which is measured in an experiment. So this is going to be your y variable. And this value does depend on another variable. So typically in an experiment, you have an independent and dependent variable. Your independent will be on the x and your dependent will be on the y. Then further, we have different categories of dependent and independent variables. We have two. We have categorical and numeric. So categorical variables are going to be ones which are non-numeric. So this could be any type of grouping or character variable. So it could be a color, red, blue, green, yellow, any type of color. It could be types of objects that you're comparing. It could be group names. It could be locations. It could be anything of this sort. As long as they're a category, it's going to be a categorical variable. compared to the numerical variable, which is going to be continuous or a nominal value. So it's going to be some type of digit or number, like 1, 2, 3, 4, or anything that's fractionalized as well. So 1.3, 1.5, anything from negative infinity to positive infinity on the number scale. So moving on to data visualization and statistical tests, I do want to emphasize the importance of data visuals. Data visuals or graphs, charts, or anything that you create help communicate your results and key metrics. A well-designed visual is going to allow your audience to make sense of your message or your results rather than reading a dense paragraph. And I want to emphasize that you're able to create a visual that is going to emphasize what you want to be conveyed in your experiment. So if you have a really significant result and you want to highlight that, creating a data visualization around this particular result will help communicate that result to your audience. One important thing that everyone needs to think about is that you really only want to use data visuals that relate to your question or your hypothesis and how they all relate and connect to each other. If you have a large amount of data, you don't want to just create graphs that have no meaning. You want your data visuals to have meaning. You want them to be related to the question. You want them to be related to your hypothesis or some particular message that you're trying to convey. And this is the same with statistical analyses. You want to make sure that you are using appropriate statistical analyses to address your hypothesis. If you have a large amount of data, you don't want to run a whole bunch of data analyses just because you can. You only want to run statistical analyses that address your core hypotheses. You don't want to try and find significance in your data when you didn't actually set out to find that in the first place. You only want to address your core hypotheses. And so moving on to some examples. This first example is going to be one categorical and one numerical variable. So the categorical variable in this scenario is going to be location, and we have two groups within location. So we have a control and a pond. And the numerical variable is going to be pH. So this is going to be on the y-axis. So if we're thinking about, this study or this data, the independent variable is going to be the location, and the dependent is going to be the y, or pH. And so what we're really focused on here is figuring out what are the differences between the control and the pond in terms of the mean pH. So what we can do is we could use a t-test in order to determine if there's a significant difference between the mean of the control and the pond for the pH value, or that response value. And what we have is if the p-value is less than 0.05, which is generated from the t-test, then we can reject the null hypothesis of no difference, and we would sort of accept the alternative. I wouldn't say that's true, but we're accepting in this scenario that the alternative would be that there is a difference between the control and the pond when it comes to the pH of these two locations. And so moving on, we're going to do another example of one categorical and one numerical. But in this case, in this scenario, we have three locations. So instead of just analyzing the differences between control and a pond, we're also going to be analyzing the differences when the river is included. So we have three locations in this case. Now a t-test is typically only used, is only used when there's two groups. When you're comparing if there's a significant difference between two distinct groups in your category. Now, in this case, we solve this if we want to compare three or more groups. we use an ANOVA. And so in this scenario, the null hypothesis is that the control equals the pond equals the river in terms of the mean pH values. And what the ANOVA does, it's going to test if there's a significant difference between these three groups, wherever they may exist. So what will be returned is an F statistic and a p-value. If the p-value, again, is less than 0.05 or whatever your significance level is, then, or whatever your alpha level is, then there is a difference that exists between these three locations somewhere in the combinations. Now, adding on to this, you can do a multiple comparisons test after the fact to determine the significant differences between each of the combinations of groups. So if we wanted to, we could run a multiple comparisons test to see the differences between control and the pond and river and control. And this will give us p-values for each of the different combinations. So this is all really going to relate to sample size and whether or not we are able to run a multiple comparisons test given the number of samples that we have collected. So with all of these statistical analyses, we want to have a very large sample size. So the number of samples that you collected, because you want to have, well, in any statistical analysis, you want to have a good representation of your data for a category or a variable or anything like this. So you want to have you want to estimate a metric that's representative of the entire population, and in order to do that, you need to have a sufficient enough sample size to calculate the key metric, the key statistic. So in this case, it's the same thing as before, the t-test, the p-value. If it's significant, it will have a level lower than 0.05. Then moving on to the next section, where we have one categorical and one numerical variable. Now, in this slide, I do want to highlight the key difference in the previous in this graph. So as you can see, data visualization can be extremely important. When we're creating visuals, we want to be inclusive to all members of the audience. So make sure you be inclusive. And in this way, you can see that the color scheme offers more contrast in this figure than the previous. So we see very distinct differences between the three groups, and not only that, but red-green colorblind individuals can actually decipher the color differences between each of the bar plots, compared to the previous slide where they would have extreme difficulty in deciphering the differences between the control and the pond in terms of the color scheme that we selected. So something very simple like changing the color scheme of your data can highlight the key, not only highlight the key differences, but also create a figure that's inclusive to everyone. So next, we're going to cover when you have two numerical variables and they're both independent. So what I mean by this is that both variables that you're measuring have no dependence on each other whatsoever. So these are two completely separate variables that should have no relationship to each other. And these are independent events. So one event happening for one variable does not influence the other event happening. So in this case, we did, well, on the x-axis, we have ice cream units sold, and on the y-axis, we have dogs walked. Now, the one unique thing about this is that since they are both independent variables, you can swap the x and y-axis variables, and they will give you the same results, and you would interpret them the same. Because in this case, we would only use a correlation test. We would use the Pearson's correlation test. And this is because we just want to see if there's a correlation between two independent events. we are not making an assumption that the ice cream unit sold has any influence on the number of dogs walked, nor are we making an assumption that the number of dogs walked is going to influence the ice cream unit sold. And so there's no evidence that this relationship exists, but we just want to see if these two independent events have any correlation to each other in this particular data set. And so in this case, our key metric is going to be R, or the correlation coefficient. And this correlation coefficient is going to range from -1 to 1, where -1 is indicative of a strong negative correlation, where 1 would be a strong positive correlation. And 0, directly in the middle, would indicate no correlation whatsoever. So if there was no correlation whatsoever, you would have plots all throughout. You wouldn't be able to have a trend line. The trend line would be horizontal, and there would be no indication of a correlation between both of these variables. Then we also generate a p-value, and that's just going to determine whether or not your Pearson's correlation test is significant or not. So moving on to the next scenario where we are using two numerical variables, but in this case we are using an independent variable and a dependent variable. So in this scenario we have the numerical variable, which is independent, being hours studied, and the dependent numerical variable being the test score. So in general we know that the number of hours studied, any given subject, the amount of time that you put in, typically you have a test score or a response that reflects that. So in this case, this study, or this data set, was trying to observe whether or not the number of hours studied reflects the test score and if there's a relationship between these two variables. So this is a scenario where you can have a correlation, highly correlated data, but we're also going to be using a linear regression to determine the R-squared value. So the R-squared value is going to indicate how much of the variation on the Y, so how much these points differ from each other, how much of the variation in the test score is explained by hours studied, or how much of the Y variable is explained by the X. So in this case, we have a trend line with so this is going to be a linear model. We have a trend line, and we have 95% prediction intervals. So essentially what we could say is, these data would be correlated. And also we show that there is some type of, positive relationship between the number of hours studied and the test score that you'll receive for the number of hours studied that you do. And this is always going to be when you have an independent variable and a dependent variable. So if we were to rotate these variables on the x and y axis, it would tell us a very different narrative. So in that case, we would be trying to figure out if the test score is some predictor for whether or not someone studied. But in this case, our data is for the narrative that, we are trying to determine if there's a relationship between hours studied and your test score. So placing your variables on the x and y axis is extremely important. And then the other key metric, along with R, which is going to indicate whether or not there's a correlation, and the R-squared, which is going to tell you how much of the variation in the y, or how much variation is explained by the x. We also have the p-value. So the p-value is going to indicate the significance of your regression, or the significance, in this case, of the linear model that we ran. So briefly, talking about what we covered today, we covered a brief introduction to statistics and why it's used. We covered some key terminology and concepts. And we covered some examples of different types of statistical analyses and some of the data visualizations that you might see. And with that, I wish everyone a great day and enjoy the Headwaters research experience.